Thank you kindly, Suha, for the invitation and for the opportunity to share some of our work with this wonderful audience. It's really, I have to say, it's been a very inspiring early morning for me. I've really been quite inspired by the other speakers, including in the business sector, and I echo Dr. Hajji's comments around how similar the sectors are, even though we in the medical sector really are not quite aware of how similar they are. And so I want to talk to you today about a specific application with this sort of burgeoning area that I think is going to really help us better control pandemics moving forward. And I hope that, I'm going to talk a little bit about sort of bringing in a little more science and a little more specifics of analytics here that I hope will, at a high level, that I hope will be inspirational and interesting to the audience. Okay, so just brief disclosures that I am a consultant to the Foundation for Innovative New Diagnostics, and I receive funding from the National Institute of Health in the US, but neither of these funds are relevant to this talk. So as an outline, I'm going to discuss first the benefits of pathogen sequencing for infectious disease surveillance. I'm going to discuss specific tools and methods that we use in this field. And I'm going to discuss data infrastructure and analytics for real-time genomic surveillance, so what we need to make that more effective. So just as a reminder that pandemics are not new, this pandemic that we're experiencing now is not the first and not going to be the last. So this is now an older slide that displays kind of death toll of pandemics since the 1300s. And you can see here COVID-19 at the front is now about 2.5 million, as Dr. Hajji mentioned. And it is, of relatively small consequence, believe it or not, despite its obviously a large magnitude compared to some other pandemics that we've experienced, including the Spanish flu, HIV/AIDS, which is ongoing, and others. And the reason why pandemics develop is because pathogens, at every level of them, be it viruses, bacteria, or other types of pathogens, are constantly evolving. Their genotypes are changing, and that results in different biology and their ability to infect a wider range of hosts. And this is really at the core of what we study in my group, and what has actually become at the core of improving surveillance for infectious diseases. So pathogens, I'm in this little cartoon here, I'm showing that pathogens, usually here are unicellular organisms. Each of these little circles represents a different cell in number one. And there's diversity, meaning that some have different genetic mutations, have different DNA than others. And because there is diversity and because of their differences in their genetic makeup, they could be better or worse adapted to certain environments. So for example, antibiotics, for example, their ability to jump hosts between infecting a bat and infecting a human. And so that diversity allows them to leap and cause infection more, potentially more severely. And so this selection that happens, which is essentially the Darwinian selection that we think about, selects for particular genotypes, in this case, the orange, that then can take over and the new generation can become all orange. And this prior generation of blue has disappeared. And for some organisms, these types of genetic mutations or changes can be transferred directly from cell to cell. We call that horizontal gene transfer. That generally doesn't happen in viruses, but it can happen in bacteria. But in the case of viruses, you could see it spread from one person to the other, and the generations of that mutant virus, are able to change their adaptation and continue to change over time. And so, what's really changed in the last 10 to 15 years in this medical area is that we are now able to generate sequences from bacteria, from humans, at really very high throughput and high scale. So whereas previously we could only study one gene at a time, one mutation at a time, and do specific typing, now we can take a patient sample and subject it to whole genome sequencing using the high throughput technologies, which have become cheaper and cheaper. So at the moment, sequencing a particular viral genome only cost a few dollars really overall. And for example, sequencing a bacterial genome costs $50. And it depends on sort of the length of that genome, because you can multiplex and put multiple samples on the same lane and generate data at very high throughput. And so when you generate the sequences, what you then do is analyze that data to get a specific mutation readout, like is this mutation present or absent? And this is the information that allows us to strain type or identify strains. And that strain information, you can then learn about the relationship between them and learn about how disease is transmitting in human populations. And as I mentioned before, this is enabled quite strongly by innovations in sequencing technology. And now that they're really being applied, not just in research anymore, in public health and in clinical laboratories. So this is sort of a picture of the oldest sequencer from 1987, the ABI prototype, which at the time cost about $230,000 and only generated 12,000 bases, which is sort of the nucleotide base ACG and T per day. And now that's increased by more than 1 million fold, and the cost has decreased by more than half. And this has really revolutionized our ability to generate this type of data and find uses for it for human health. And, and there is increasing technology to kind of miniaturize this type of sequencing platforms. And this particular sequencer, it's called the USB sequencer or the MinION, literally fits in somebody's pocket. And, and it's really quite impressive to see these demos of these sequencers that can be carried everywhere and can be used even in the most difficult or, least in settings where there's very limited laboratory capacity. And famously, this technology was used for Ebola transmission tracking in West Africa in 2014. So then thinking about how you analyze the data and how you kind of glean knowledge from it and interpret it. So at a very caricature level here, just to make it simple, when you have different strains, you're sequencing them and you identify specific mutations. And for example, in this case, the yellow and the orange strains have the same base at the site, and the blue strain has an A. It has a substitution in that site. And so immediately you know that the yellow and orange strain are closer in genetic distance, all else being equal, than they are to the blue. And so you can kind of essentially construct what we call a phylogenetic tree that essentially represents the relationship between these different strains in this manner. And this tree is very useful. And and I will give you an example of how it's used in real life. But I think underlying all of this is the fact that there is a molecular clock that usually represents most living things, most genetic sequences, meaning that over time, mutations develop. Most of the time, they're completely random and have very little consequence on the phenotype or the ability of that organism to cause disease. And it ticks at a specific clock. And, there's a specific rate by which mutations develop. And this is because essentially the replication machinery of that DNA is faulty. It's not perfect. There's errors that happen at random. And so if I'm showing you here a plot, it's called a root to tip regression, where I'm showing you the distance in SNPs on the y-axis, mutations. So the count of how many SNPs develop from a root, from some reference or original viral sequence. In this case, these are all COVID-19, SARS-CoV-19 sequences. And on the x-axis is time. So you can see that if you count SNPs in these hundreds to thousands of isolates that we have here, which is I think approximately 4,000 total isolates, you can see that the mutations over time is roughly linear, and the rate is approximately 25 mutations per year, meaning that SARS-CoV is mutating, accumulates one mutation every 2 weeks, and this is the expectation. And so knowing this, you know that because essentially mutations represent time, you can then understand transmission, meaning that when you see mutation, you see two genomes isolated from two different patients that have no mutations, you know that that viral strain has actually transmitted in the last 2 weeks, more or less, between those patients. And so when you see larger distances, you know that there's a lot of intervening infection that you haven't sampled. Or if you see very, very large differences, you can infer something like importation from another country, where this is the first case that you see in this country. And so this is incredibly useful for public health. To just give you a further example, so when you build a phylogenetic tree, for example, if you have this, you end up with this scenario where you have your one isolate and you're comparing it to isolates from other parts of the world. And you find that there's only one isolate in Lebanon that has that genotype, and all others were found in this other foreign country. And this is an example of an introduction or importation of that strain into Lebanon. But in this other circumstance, when you've sequenced adequately and you find that there's multiple of these strains in Lebanon, you can be sure that there's ongoing community transmission of that strain in Lebanon. Of course, things are, I'm simplifying them here. There's more subtleties, and again, happy to take questions. But I wanted to give you an example of an analysis we undertook at the Massachusetts General Hospital here in Boston that investigated SARS-CoV transmission in our state. And this was a very large effort led by the Broad Institute, and I played a small part in it, but I'm really happy to be able to share with you some of these results. So very early on so Boston was very heavily hit, and it was hit very early. And it's because of how connected our city is to other parts of the world, especially to Europe. And I'll show you how we actually gleaned that from the genomic data. So we sequenced approximately 800 genomes from essentially from the lab at Massachusetts General Hospital, but also from the Massachusetts State Public Health Lab that also provided samples for sequencing. And then we studied the genetic relationships of these strains and also studied importation in and out of the Boston area. So this first panel here demonstrates the sampling strategy that initially the sequenced isolates, which are represented in green, and the overall isolates represented, the number of positive cases are represented in blue. You could see here that at the beginning of the outbreak in Boston, we have essentially 100% coverage of what was circulating in the state. Later on, we couldn't keep up because the SARS-CoV was spreading and spreading in multiple sites around the state at where patients were seeking hospital at other centers, and so our coverage of the sequences dropped. But we have very good coverage very early on in the outbreak, and you can see here where the samples, where our patients came from, as represented by this map. And Boston here is in the middle, and where obviously it's a large urban center. And where the population density is highest. And we had actually some particularly hard hit neighborhoods in our city that are very well represented in our sequences here. And so, this is the phylogenetic tree. Hopefully now it's not too foreign to you. Of all the isolates, all the red isolates are from Massachusetts. All the other isolates are colored differently. All the isolates from Europe are pink. And you can see here that you can very easily see that most of the importation and spread into Massachusetts came from Europe. So the neighboring isolates were all pink, essentially. So the context is predominantly pink. And so I'm going to focus specifically on one particular group of isolates here that are marked by this mutation, C24160. And essentially, this is these sets of mutations and these strains were first found in individuals who were infected at a business conference that happened in late February in Boston. And so actually, interestingly, this genotype and this particular strain was a predominant, it constituted the grand majority of strains in Boston early on in the pandemic. It did then we did get more diversity as more importation happened, because we estimated a total of approximately 120 importations of SARS-CoV into Boston at that time period. But it did actually turn out to it definitely continued to spread within the Boston area. We found it within homeless shelters. We found it across the state. It actually spread to 19 other states in the US based on essentially the surveillance, the sequencing surveillance that's happening in other parts of the country. And it actually then spread to Australia and other parts of the world. And so here, this essentially phylogeny demonstrates this, that some of these blue isolates were first found in Massachusetts. This new mutation developed within this larger group and spread locally and globally, and all these gray isolates here represent global spread. And so you can easily see, you can track essentially where these strains are going and how much they're infecting. And it's estimated that upwards of 300,000 patients have been onwards infected from this essentially sequential transmission. And this is a map of where all these strains were found in the US. Okay, there are other applications to pathogen sequencing, including surveillance, as I was discussing, but we also can predict, for example, antibiotic resistance, and this is very important information for the clinic. And we can understand other forms of phenotypes. For example, since learning about the diversity that SARS-CoV has demonstrated, we now know that certain strains are more transmissible and have a higher case fatality rate, including the example of the UK strain. There's also the South African strain and others that have been found since. And the reason why we have been able to identify the UK strain as more transmissible has everything to do with the surveillance that's happening in the United Kingdom and how extensive and exhaustive the effort there has been, where they've been sequencing upward of 10% of their daily samples, weekly samples that turn positive. And that's really enabled the systematic evaluation of transmissibility, of case fatality rate that otherwise would not have been possible. So as another, to kind of change flavors a little bit from SARS-CoV, to give you another example of how genetic mutations can affect the phenotype or the virulence or transmissibility of a particular pathogen. This is an example from Ebola, where, as you can see in this phylogenetic tree, initially all of the isolates, all the Ebola isolates had this genotype allele A at genotype site 82, and over time, there was a substitution from A to V, and all subsequent isolates that were sampled and sequenced in this kind of global effort have all been the V allele. And in fact, we know for a fact that it's sweet. So meaning all of the daughter virions had developed the V allele. And in some basic science experiments that were performed, it was comparing the wild type, meaning the A allele carrying virions, to the V allele, the mutated strain. It was demonstrated that actually these virions with the mutated V strain were more effective at causing, essentially, they replicate more effectively within human dendritic cells, and that essentially is evidence supporting that it's more transmissible from one patient to the other. And this has also been demonstrated nicely for some of for isolates, including the UK strain in the literature. So beyond viruses, there's also a lot that we can learn from for bacteria. For example, in this study, we used a very large data set of TB isolates and actually were able to reconstruct when antibiotic resistance developed in different parts of the world, and we're able to infer that there is actually a order in which antibiotic resistance develops in TB, including to isoniazid first, and then rifampin and streptomycin, and then the other drugs. But this was very interesting because it taught us that most of the streptomycin resistance that's circulating around the world developed around the time rifampicin resistance developed, even though streptomycin is a much, much older drug, and it's the original antibiotic. And so this actually taught us that it was the practice of adding streptomycin to a failing treatment regimen that actually resulted in amplification of streptomycin resistance, and not the age of the drug introduction. And this is really, it's essentially a forensics of public health, and the forensics of what treatment strategies we need to employ to better care for our patients. And in other work we have done using a different metric that actually allows us to get to this interesting assessment of transmissibility. We find, and so here in specific, we look at phylogenetic trees and we measure specifically the terminal branch lengths, because when you have a faster, more efficient transmission, these terminal branch lengths are shorter. And when there's less efficient transmission, they can be longer. And essentially, we demonstrate here that for lineages 4 and 2, which are two specific types of strains of TB, there is shorter branch lengths, and the p-values here are very, very tiny compared to these lineage 3 and lineage 1 isolates, demonstrating that there are strains within TB that are more transmissible, are more efficient at communicating between patients than others. So this is not just a phenomenon restricted to viruses. And when we pool all of this data together, along with phenotypic data, specifically here, antibiotic resistance profiles, we can then harness the power of modeling and statistical learning, or machine learning, if you will, to predict the phenotype of the organism from its genotype. We've done a lot of work in this space. This is just one example where we were able to predict antibiotic resistance with very high AUCs, upwards of 0.9 in most cases, using this approach. And this is really empowered by data, by having the sequencing data, represent countries around the world, and then so that these models can then be used and ported and applied in different parts of the world with high accuracy. So now I'm going to kind of conclude with a few slides talking a little bit about data infrastructure and platforms. So please let me know if I'm kind of running over or getting close to the limit.
2 more minutes.
Okay, thank you. So this is I put up this picture of the GISAID website because this has really enabled quite efficiently the ability to study SARS-CoV strain variation. So this is a platform that was initially built for influenza that has become a hub for international data on SARS-CoV genomics. And it's really staggering because now there's over 700,000 SARS-CoV genomes. We have, for the first time in my career, more data that we can analyze. And it's really quite incredible. And this is in part related to the investment in many parts of the world, especially UK, US, well-resourced countries, but also in other countries. There's really an incredible amount of investment of sequencing in Africa. There's over 130 countries represented. And it does sadden me that the Middle East region has actually very, very limited representation in this forum. But ultimately, for this data to be effective, we have to adequately and systematically sample it at the regional level, at the country level. We have to link it to clinical and epidemiological data, such as the data that Dr. Hajia mentioned earlier, as well as be able to analyze it in the context of the global data so that we can contextualize and understand transmission and survey diseases better. And there is an ecosystem of analytic tools that has evolved around sequencing, including, the poster child, which is nextstrain.org, which is really an incredible effort around benchmarking and best practices of how you analyze sequence data, how you build these phylogenetic trees accurately, how you quality control the data. And they've really been really great pioneers. So shout out to them and their great team. They have actually, everything is open source. People are building on their work. They're downloading their work. It's really been quite energized, this space. And so I don't have to say, tell this audience, but improve, democratizing and open sourcing analytical tools helps us improve accuracy, reproducibility, standardizing outputs and naming conventions, and facilitates communication and action. But I also want to say that there is something to be said about building tools that are pathogen specific, because there are often phenotypes and data considerations that are pathogen specific and they simply don't apply or wouldn't necessarily be developed. And so we've built a tool for TB around that concept and predicting antibiotic resistance, where people can upload their data and get a prediction, again, in the spirit of democratizing and disseminating analytics. Again, building on open source software whenever we can. So in summary, pathogen whole genome sequencing can answer important questions relevant to public health and infectious disease management. Understanding local strain diversity is paramount to proper control measures. Some examples include, for example, prioritizing travel, so versus local lockdown, understanding healthcare associated versus community transmission, identifying and responding to vaccine escape, which we're already starting to see with some of these UK strains and the South African strains. So surveying what strains are continuing to cause disease as we roll out vaccines should be a priority. And finally, we need investment to make this a reality for many countries around the world. And that so far, data sharing and analysis tools are well developed in this space and will continue to be refined over time. So I just want to thank my team and our funders. And this is us in the Zoom era, celebrating with our kids and our drinks when one of our team members here, Jessica, moved on to her next job. And it's really quite a privilege to work with this wonderful team. And I'm just going to leave this slide up here for a few seconds as we take questions. This is a nice review article that talks about what we need to do to operationalize molecular type surveillance in resource-limited settings. So thank you all for your attention.
Thank you very much, Maha. This was an outstanding presentation. And you know, as you have talked about in details, this is a tool that we're using increasingly in investigations of various infection control outbreaks, and we've used it to investigate outbreaks of Acinetobacter at AUB-MC in the past. We're currently using it to investigate an outbreak of Candida auris, a fungus we've never seen before at AUB, and you gave us a very good example of how this can help with the understanding of the transmission of the COVID virus as an example. I think, I'm sure there are many questions, but also to try to keep track of time, we're going to skip the Q&A because also Dr. Farhat is going to be one of the panelists, and I'm sure she'll be happy to answer your questions. Please type them in the chat box, and she'll be with us again in the second session. Thank you very much, Maha.
Thank you.
The last presentation in this session is by Dr. Carla Talarico, and I have the pleasure to introduce her. Dr. Talarico is a senior director and lead epidemiologist for Moderna COVID-19 vaccine. She also serves as the lead epidemiologist for other vaccine development program, such as CMV, EBV, and others. She's also building the epidemiology department at Moderna with an ambition to create a world-renowned group of scientists focused on observational research, epidemiological studies, and real-world data and analytics. Carla received her PhD in epidemiology and MPH in epidemiology and international health from the University of Michigan, Ann Arbor. Before joining Moderna, she worked initially at the US CDC, where she served as a subject matter lead in microbiology and molecular biology. And after that, she worked in JSK, where she was heavily involved in the development of studies for pneumococcal vaccines and the varicella-zoster Shingrix vaccine. So Dr. Carla is going to talk to us on big data to help a big problem. Use of real-world data and bioinformatics to show effectiveness of COVID-19 vaccines and track SARS-CoV-2 variants. Carla, the microphone is yours.